Hey, good morning, everyone. Thank you, Keertana, for this wonderful introduction. So, welcome you all for this summer internship in DNA sequence analysis. Why this internship? Barcode biotechnology. We've been facing this dilemma for more than 5 years now. We need more hands for analyzing DNA sequences. Once we generate Sanger sequencing data or Illumina data, we need more hands for analyzing the sequences and preparing the report. Here is when it's a bottleneck in the DNA sequencing services. If this one step usually gets delayed, because of which we may not able to deliver the results on time to customers. Okay, so every time we train a batch of students, then we hire them, but we couldn't retain them for various other reasons. So this time we thought that we will generate an internship program and intentionally train what is required in this industry in terms of summer internship and hire them back once they are skilled. That's an idea behind this, but okay. What is this barcode biotechnology? We founded in 2013. We are basically a DNA sequencing service providers. We provide Illumina and the Sanger sequencing services. Actually, we started with Sanger sequencing services. As the name indicate, we initially provided services for molecular identification. Now we gradually increased our services for mutational analysis, diagnosis of pathogens from food and environment, and many more. Okay. We are a small team of 35 people involved in primarily in DNA sequencing based services. Okay. So I was told to project the infrastructure. So we started with a megabase 1000 sequencer with 36 capillary tube sequencer. This is how we started with, and we later on procured ABI sequencer 3700XL. And then we recently installed Illumina platforms for next-gen sequencing. We have a PCR workstation that can handle around 700 samples, in 3 hours time. And we have a bread provider, Mermaid 192X synthesizer. This is a primer synthesizer. It runs 24/7. And they are actually a bread provider for the company. And these are the various sequencing services we provide. We provide, like I said, gene-based sequencing services. We provide a gene expression identification system where we isolate your RNA. We go for cDNA synthesis, targeted prime PCR amplification, and then sequencing. We also provide microbial, animal, plant identification services. Even if you want to identify your bacteria very precisely, we recommend multi-locus gene sequencing, where other than 6' RNA gene, RNA polymerase beta subunit B is being targeted. Okay. So we also provide meta-barcoding services. You can just give us a community sample. We will tell you what are the animals there in the community. Okay. It need not be an isolated species. We can decode one gene at a time, and we'll find out what are the animal composition in the community. Other than these services, we provide internships. We are also hiring bioinformatics associate who we expect to handle the skills we are going to impart during this internship services. So, starting with this internship, what is this DNA? The barcode after the name barcode in the company actually indicates the universal barcodes in every product, where just by reading the universal barcode you can decode the product. Likewise, reading the DNA sequences, you can actually find the animal species name or plant species name. Hence the term barcode was given. This is actually given by a professor from University of Gulf, Paul D. Hebert, in around 2009. From there, we also got initial funding from them to initiate this company during that period. And finally, by 2013, uh, we have crossed the turnover point, and we've been in business. So whenever we are talking about DNA, we couldn't forget these 5 experiments. Griffith experiment, which initially proved that DNA could be a transforming principle between the rough and smooth strains of Streptococcus. Then the experiment has been proved by Avery and McCarthy, where they clearly say that DNA is the genetic element that is being transferred. Hershey and Chase also complemented this finding after that. And when Watson and Crick came up with this structure of DNA, things have been settled down. Finally, the measles and Stahl has proved how the DNA replicates in the semi-conservative mode. So, what is the significance of sequencing a DNA? You see now, DNA sequences has become a gold standard for species identification. Doesn't matter you have an E. coli for 5 years, you are working with that, you are publishing papers, but unless until you provide a genetic data for that E. coli to prove it is Escherichia coli, scientific community may not be accepting the data. So it has become a gold standard that you sequence a DNA for species identification, for disease diagnosis, and for many purposes. Even for proving your antibiotic resistant, you need to show them that that particular antibiotic gene is being expressed through RNA isolation, cDNA synthesis, and target PCR and sequencing. So how this barcode works? Barcoding is nothing but, like I said, finding a species name. Okay. So once you collect the sample, here is an overall picture. Once you collect the sample, go for sequencing, generate barcodes, deposit in the reference database like NCBI, DDBJ, where you will just give the sequences under a species name. The all known species that can be easily identified, you are actually sequencing. You are submitting the sequences with the species name. Okay. So once such a database is created with thousands of sequences, now any unknown sequence can be taken and compared to the database, and you will find the species name. That's how barcoding works. That's how identifying a species using DNA sequences works. First, create a database with all known species, and once you have unknown species, or new sequences from unknown species, you can just compare it. Yes, another disadvantage of this technique is your local library, the library you built, the barcode library should be encompassing of all species. Only then it will work perfect. Any unknown sequence can be given. So what if I couldn't or we couldn't? Still, it's a problem. They couldn't generate all species in the library. We will see how to tackle that problem using the terminology called barcode gap and others. Okay. So, like I said, this is how the DNA taxonomy works. You take a known species, sequence it, deposit in the library. So that library will have millions of sequences of known species. When you take an unknown DNA sequence and you query against it, uh, you will come to know the species name. So this is the entire workflow of the barcoding, where you will sample the animal as plant or microorganism, whatever it is, isolate DNA, go for PCR. After isolating DNA in the PCR, only targeted gene regions are amplified. See, for plants, they have a Maturus K or ribulose bisphosphate carboxylase large subunit L gene as a DNA barcode. And for animals, we have COI, cytochrome C oxidase 1. And for bacteria and prokaryotes, archaea, we have 6S RNA gene. And for fungi, we have internal transcribed spacer region, ITS. So all these regions, they constitute as DNA barcodes. So once amplification is done, two-way sequencing is done, and from that sequencing, the data are deposited in databases like NCBI, or also a dedicated database for barcoding is Barcode of Life database system. So our internship will be focusing on what happens when the sequences come out of the sequencer. So this is the overall Sanger sequencing workflow where, this is the theory, I guess, this, we denature the DNA and deoxyribonucleotides are added to that. Okay. And we will be adding the ingredients of DNA synthesis, adenine, guanine, thymine, cytosine. These are the bricks of this building. And from that, since we have added deoxynucleotides, there is a intermediate strand termination, and after that, these strands are run inside the gel capillary electrophoresis, and the fluorescent molecules will direct each and every deoxy molecules that is been passing through. That's how we will create a chromatogram. So I know this is a basic principle. I just want to refresh your memory before we get into it. Okay, that's how these chromatograms are created. So DNA chromatograms are machine output files during Sanger sequencing. Okay, there each and every peaks represents every nucleotide. Okay, so our internship will start with how to handle the chromatograms. Why to handle? I will tell you why. You might be surprised to see other than ATGC, there will be WOS or those kind of codes in the DNA sequences. So here is an IUPAC table. Say, for example, if you have a code W, it means it can be A or T, meaning the machine couldn't call the particular position during chromatogram synthesis. When I say position, for example, here in this gene length, 120 represents the 120th base pair. The adenine here is a 120th base pair. So whenever you see R, the machine couldn't call whether it is adenine or guanine. Whenever you see, say for example, N, that can be any base pair, any of the four base pair. Okay, so this is an IUPAC code you need to know. Many times I have, in my experience, I have seen this code can be reversed by manual inspection of the chromatogram. So that's where we will start our internship. We are going to manually inspect the machine output file, that is DNA chromatograms. Okay, so we call them as a degeneracy. These codes, they can be used in various ways. For example, if we don't have example D code, which can be adenine or guanine or never thymine. Okay, so this code, if it is not there, what do you do when you generate a DNA sequence? Can we keep on taking notes like position 121 could be adenine or guanine or thymine? For how many positions you can write such an instruction and keep it along with the DNA metafile? It is something very laborious. Okay, so these codes are actually compact and it's very informative, and they are widely recognized in bioinformatic sequence analysis services. Okay, so here is our entire workflow of our internship. We will start with this is not in the exact order of how we are you going to do, but this is something we are going to do. We will learn how to analyze a machine output file with actual examples from the barcode sequences. We will construct extract the sequences, and we will go for consensus generation. I will tell you what is this consensus. And we will do a blast analysis to find what species it is or what gene those sequence belongs to. After blast analysis, we will learn how to do multiple sequence analysis. Okay. Any bioinformatics analysis will start with alignment. Right. So whenever you handle a DNA sequence, it's very important you align them before you subject them to further analysis. So the second thing we'll learn after alignment is genetic distance data generation. We will compare the DNA sequences. We will try to analyze the distance data between the DNA sequences. Finally, with these sequences, we will generate a phylogram. Okay, this is how our internship, skilling part of the internship will progress. So I will just touch upon little things about what is this manipulating a DNA chromatogram. Okay, see, as you can see here, both the peaks are of same height. This can be either A or either T. Okay, so you need to check the IUPAC code. If it is either A or T, this can be a W position, not the T position. Okay, so likewise, there are noisy chromatogram, how to identify, how to handle, how to edit it. We will go through that one by one. So why to edit the chromatogram? The chromatogram you're seeing here is an exactly a perfect chromatogram, but not always all chromatograms will appear like this. Okay, so how will we do that? We will install a software called Chromas Pro. We will import the machine output file into it, and I will tell you how to edit those chromatograms. Okay. Once you have edited, you can extract the sequence out. So see the even the Sanger sequencing. they cannot, the fidelity of tag polymerase cannot go beyond 1000 base pair, 1050 is a maximum length of a gene where a continuous synthesis can happen. But this is a problem. For example, the 16S RNA is 1500 base pair. Okay. Once you do not generate 90% of sequence length, most probably your sequences will not be useful. When I say it is not useful, when you subject them to blast analysis, say for example you have an Bacillus subtilis sequence. When you submit them to blast analysis, it will say if it is less than 90% length, they may share 100% similarity with Bacillus series and Bacillus subtilis in the blast analysis. Now, what species it is? Both are sharing 100 species similarity. Your purpose of sequencing is ruined here because it's not identifying as a one single species. It is showing percentage of. So this kind of problem will come if the sequence length is less than 85%. So in barcode biotechnology, they take a special care to produce 90% minimum of 90% length every time they do. So how do they do this? So the like I said, at least 900 base pair at one single stage can be produced. So both the strands must be sequenced. Two-way sequencing should be done for every sample, both forward and reverse. Forward will be 900 base pair, reverse will be 900 base pair. Now we can reverse complement the reverse and fuse these two strands, and we can extract 1500 base pair out of it. Okay, that's how it's done. We will have this practical to how to compile those forward and reverse strand after chromatogram manipulation and go for extracting that. Okay, this is what I said. So, for example, here we have a forward strand here and we have a reverse strand here. When we fuse it, the length of the strands can be improved. Okay, so this is the basic principle behind two-way sequencing. What is this blast? Yeah, it's an online database. Imagine they have the sequences of all known species. Once you want to know what is your species name, usually you take the sequences, you query or you search the database. Okay, but yeah, this is like Word document. Control F. You try to search the name of some employee in your database. Just write down their employee name and hit enter. If it is there, they will strike it. Right. Likewise, blast analysis usually take your query sequence, compare with the database. If it get the hits, they will release the result. We will see what is this alignment is doing during searching your query with the database, how these species names are declared. All those things we will come back and we will see them in detail. Okay. So genetic distance analysis, finally, once you know your species or once you have a doubt whether it can be this species or that species, genetic distance analysis can give us exact results. We can take a numerical data driven decisions based on genetic distance. We will collect the reference sequences from the NCBI and we will use your own sequences to calculate the genetic distance analysis. Okay, say if two sequences considered as they have a very dissimilarity of 2%, what it means is when you compare 100 base pair of one sequence with another sequence, you know, the two base pair may be different, so it is 2% variation. If it is an 1000 base pair sequence, so 20 base pair variation. Okay, so likewise, percentage of variation usually indicates the number of base pair that is being differing between two nucleotide sequences. Okay, for 16S RNA, for long, the accepted barcode gap or species identity gap is 3%. Meaning, if you are querying E. coli sequences in blast analysis, if the sequence shared a 97 or 98% similarity, it is usually E. coli. But if it is shared 96% similarity, it is Escherichia genera, but not coli species. Okay, so this is called species gap. For every species, we need to identify we need to know the species gap, which is a challenging task right now in bioinformatics. Even there are, I can point out many domains, Nidarians and many domains in biodiversity where they don't have exact species gap. Once this environmental barcoding and other barcoding services, which is already picking up, generating millions of sequences. If the local library do not have DNA sequence, do not have species gap, then they are in real trouble because this technology may not be fully functional if these data are missing. So the second objective of this internship is to train the students in genetic distance data analysis and phylogram construction, where they can identify the phyla that don't have a species gap or a species that don't have a gap and choose it for your mini project. Yes, we will be having a mini project here once the scaling part is done. We will make some technical report and see whether we can put it in our yeah, scientific platforms to to advertise your skills. So what is this genetic phylogenetic analysis? Distance data is a numerical data. Phylogenetic is a representation of those similarity and variation in graphical form. Okay. Say for example, from this phylogram, all phylograms should be observed something from left to right. I could say this A and B are closely related when compared to A and K. They are distantly branched out. Right. So not only genetic distance, even phylogram can be used to track the source of viruses. Even in the recent pandemic, that's how we did it. We know it is from bad origin that has been came to humans, the coronavirus, just by constructing the phylogram, the whole genome of the virus. So phylograms are something can be used to track the ancestry, cross species transmission, okay, and diversity of particular species in different hosts. All those things can be analyzed. So that's an objective of phylogram. So our internship roadmap will be skilling you in these analysis, machine chromatogram manipulation, blast analysis, how to calculate a genetic distance data, how to draw a phylogram, for which I request every participant to choose your own species. Okay, choose your own species for a mini project. We can work from now on. All right. It can be a microbial species, plant, animals, virus, doesn't matter. I know we have students from botany background, zoology background, life science, uh, many diversified background. You choose any species, any one biological species on your own. I will circulate a Google form to get the species name from you. From there, each and every participants will be focused individually, and a mini project can happen from there on. Okay. Finally, once your analysis is done, I expect I will tell you how to choose the species and other stuff. We will choose a species that do not have a barcode gap. Okay, for which the barcode gap is not available in the database. First, you choose. Let me do this searching and see what are the species that do not do the barcode gap, or I will also circulate the list of species that can be chosen which do not have a barcode gap. And finally, we will derive the barcode gap by these analysis, by genetic distance analysis and phylogram. We will propose a gap for that species. We will submit that proposal in form of a technical report and we will publish it. Okay. So not only you will get skilled in all these assays, we will also advertise to the world that this after publication of the technical report that this skill has been successfully illustrated, you have been successfully skilled. So that's all for now. Today it's an orientation session, so we thought this is what we are going to do. So for this, I would advise attend your Google Meet every day through your phone. The link will be shared in the group, so it will be easy for you to attend the meet through your phone, and parallelly keep a laptop connected to internet. Okay. So whatever you are listening, seeing, and hearing from the phone, I would say parallelly you practice in your laptop. That's how bioinformatics can be done very efficient. All questions are welcome. This internship will be of in both the modes. You will have a recorded video shared someday, and you will have a live session shared someday. You can message me, or you can send your queries to the mail anytime. I will be happy to help you with. Okay. Every day, recorded sessions or the live sessions will be shared from 10 o'clock. There will be a little assignment. We'll see how we can find a barcode gap for undefined species. So that's all for now. Happy learning.